Hey, everybody. Today we're going to return to the idea of some probability in genetics, and we're going to look at how we can use a new test, the chi-squared goodness of fit test, in order to test hypotheses about the genetic makeup of our population. So there's a couple different applications for this, and so let's get started. All right, so just to review, we're going to have categorical data. That is, we have some individuals that we've looked at and we have counted up how they fall into different categories. So this is an example of pet ownership in US households. These data, assuming we had 500 people, these numbers of individuals get translated into percentages of the population for us to put them onto this pie chart over here. So 65 out of 500 represents 13% of people we surveyed were never a pet owner. Current pet owners were 335 out of 500. That makes 67%, all right, by far the biggest pie wedge here. And then people who had a pet previously but don't have one now are the rest of the individuals. So 20 out of 500 is where we get this 20% from. Right, now we can break down these current pet owners by what kind of pet they have. So split out is a little bar chart showing out of all the current pet owners, what kind of pet do they have? Half of them have dogs, a third have cats, et cetera, et cetera. So we aren't showing this on a pie chart because this adds up to more than 100% because some people have more than one pet. All right, but we're going to take, using these percents, I found the number of individuals out of our sample that had each type of pet, but we're not going to take it out of 500 because we're only looking at this current pet owners pie wedge. So those were 335 individuals. And when we break it down across all these different pet types, that means 167 had a dog, which is 50% of those 335. Right, I skipped a few columns here because you're smart and I think you can see how the pattern works. All right, so we have 3 different categories for our overall pet ownership. 1, 2, 3. The sum, remember, it doesn't count as one of the categories. And then we have 1, 2, 3, 4, 5, 6, 7, 8 categories that we're putting our different pet types into. All right, and I don't have 8 columns here because I didn't want to try and squish them all onto the screen. Right, so we can see how many columns we have in our table is also going to tell us how many categories we have, right? So what questions we might be interested in asking when we have categorical variables like this is there a difference between our distribution? So our distribution is how those individuals are separated out across those categories. So we might ask a question, is there an equal number of people who own dogs and cats, right? So in this example, we have these red bars representing something and these blue bars representing something else. And each of them are shown for, say, maybe these are an age category, and maybe these are proportion of individuals that use social media and don't use social media or something like that. Are they distributed equally across these different categories, or are these two distributions the same as one another? Now, just from looking at this example, the answer to either of those questions would be no, but sometimes it's much closer, right? And the first kind of statistical test we're going to look at is going to use these differences in the heights of the bars here. And we're going to call one of our sets of bars, we're going to call them observed data, and we want to compare it to some other set of data that we expect because of what our null hypothesis is. Right, and we're going to be able to do hypothesis tests on this kind of data because there is a chi-squared distribution, and so we call the statistical test we use the chi-squared test statistic for, that is going to allow us to compare how many individuals we have in our observed group and our expected group in each of the categories. So as we look across the different categories, we want to summarize. In this case, we're literally just going to sum up the square of these differences divided by the expecteds. All right, so we're going to call the number of categories we have little k. So we'll have, if we have 3 categories, we'll say little k is 3. We have 8 categories, we'll say little k is 8. And one of the assumptions for this test, about the only assumption for this test, that we have at least 5 counts in each of our categories. So it works on pretty small data sets. All right, so calculating how different the observed number of individuals is in each category compared to the expected number of individuals, right, based on our null hypothesis. We're going to calculate this test statistic, sometimes capital X, sometimes it's called chi-squared, but it's an estimate of a chi-squared. So we're going to take the number of observed for the first column, subtract the expected for the first column. We're going to square that quantity, and then we're going to divide by the expected. We're going to do that for each of our columns, and we're going to sum those up. All right, so the bigger the difference between the observed and expected, and the more we add up across all of the columns of a table, the bigger our value of our x or our chi-squared is going to be. So this graph is showing an example of the chi-square distribution, what it looks like. It's not like the normal or the t-distributions that are symmetrical. It's asymmetrical. It has a long tail out to the right. And our values of chi-squared are always going to be positive because when we do this subtraction step, we're squaring those values. All right, so the bigger the difference is, the bigger our value of chi-squared is. And this figure from the top of your chi-squared table, chi-squared distribution table, is showing us that the probability little p is the area under the tail of this distribution to the right. And we're only ever going to look at the tail to the right. So we want to know what's the probability that we get a value of chi-squared as big or bigger than the one that we got due to chance alone if the null hypothesis is true. All right, so the bigger the value of chi-squared, the smaller the probability is. All right, so the bigger the difference between the observed and the expecteds, the less likely we believe our null hypothesis is. All right, and logic as we've used in all of our hypothesis tests so far. All right, the tricky part about doing chi-squared tests is they're a family of tests. We can do them on several different experimental situations or research questions we want to ask. So this expression, the observed minus expected squared over expected, is always true. That's always going to be the equation. But depending on the application we're using, what the observed values are and what the expected values are is going to depend on the type of test we're doing. So today we're going to start with one of the kinds called the chi-squared goodness of fit test. We're going to do a chi-squared test for independence in the next lecture. And what we use for these observed and expecteds, primarily the expecteds. The observed is going to be what we found in our sample. Right. The expecteds are going to depend on what the null hypothesis is, and that differs depending on type of test. Right. So you have to think through the logic of how this test is going to work and decide how to calculate the expecteds based on experimental design and the type of test that you're doing. Right. Now, there's only a couple categories that we'll learn in this class, so it will be relatively straightforward. All right, a couple more things about the chi-square distribution. Like the t-distribution, it's going to depend on the number of categories, the shape of the distribution, right? As we add categories, we're going to add that chi-squared value is going to get bigger and bigger. So we want to take into account if we have more categories or less categories. And the way that we do that is that the chi-square distribution is going to have a different shape. Now, it almost looks like a negative exponential when the degrees of freedom is very small. When the degrees of freedom is larger, it's going to get less and less right skewed. And if we have 1,000 or more degrees of freedom, it's 1,000 categories that we've put individuals into, which is not very likely for an experiment, but just to dot our I's and cross our T's, how does this relate to the normal distribution? If we have a very large degrees of freedom, like if we had a very large sample size in our T distribution, it's approximately. All right, the degrees of freedom are going to depend on K. So little k is the number of our categories. The degrees of freedom for most of the tests will be k minus 1, but it depends a little bit on the type of test. All right, so we'll have a different way to find the degrees of freedom depending on the test. Here's what your chi-square distribution table looks like, the very top of it. Now, we're typically only going to be using a very small number of degrees of freedom because we'll have a small number of categories in the types of data we're looking at in this class. So this is the very top of the table. It goes from degrees of freedom 1 down to 10. And what the table is telling you, the chi-square distribution critical values, is that there's values of chi-squared actually in the table. Right, so notice they start with a degrees of freedom 1. The value of chi-squared is 1.32 here on the top left. As we go across the row to the right, it gets bigger and bigger up to 12.12. Right now, if we go down the rows, as the degrees of freedom gets bigger, the critical values get bigger as well. All right, so as we go down, as we go to the right, the values of chi-squared will be larger. Now, what at the very top of each column is the p-value, right? So if our chi-squared is exactly 1.32, our p-value will be 0.25. Our chi-square value has to get all the way up to 3.84 for us to be at the 0.05 p level, right? So if we have a 0.05 alpha level, our chi-square value, if we have 1 degree of freedom, needs to be bigger than 3.84. So this distribution works very much like the t-distribution, where we have the critical values inside the table, values of chi-squared. We're going to find our value of chi-squared for the correct degrees of freedom in the table. If we're in between, then we'll go up to the top and say our p-value is in between those two values. All right. The other thing is if our chi-square value is bigger than in this first row, if it's bigger than 12.12, that means our p-value is going to be less than 0.0005. All right, same thing at the left-hand end of the table here. If our chi-squared value is less than 1.32, and if our chi-square value is real small, that means there isn't a very big difference between the observes and the expecteds. right? So our p-value will be less than 0.25. That means we will not reject the null, right? So small difference between observed and expected is going to give us a small value of p, a large value of p, and that means we're not going to reject the null, right? I'll work through an example in a minute. All right, so what is this test, this chi-squared goodness of fit test? What does that even mean? All right, so I like to think about goodness of fit with an example that many of you may be familiar with, and that is you go to the store, you try on a pair of pants or a pair of jeans, and you have to ask this question, does it fit? All right, does it fit? All right, so we have some categories that we have to compare our body, our perfect, beautiful body, to these stupid jeans that somebody made in a particular size. All right, so we're going to have a waist. Does its waist match our waist? A hip, or is it big enough in the butt? Thighs, maybe the style. Is it, you know, does it make you look tall enough? Does it make you look short? These pockets make your butt look weird. Are they in the right place? Are the calves too tight? That's really aggravating. All right, so each pair of jeans or of pants, whatever, it's going to be too tight in some area, too loose in another. And if we want to add up all of those, sort of like, how different is it? Does it fit? All right, we're going to have that fit across all these categories. Now, if you're going to take those perfect jeans home with you, all right, they're probably not going to fit exactly perfectly, right? But depending on how long you've been shopping and how frustrated and tired you are, you're going to have a certain amount of difference from your beautiful, perfect body that these stupid pants are, right? And so it's going to be close. Maybe it's a little bit too big through the thighs, but you're willing to take it, all right? So there's going to be small amount of lack of fit that you're going to accept, all right? But if it's too different, all right, if everything is perfect except the waist is 3 inches too big, all right, you're going to reject the null. Those pants don't fit you. All right, so in the goodness of fit test, our null hypothesis is going to be it fits, they're the same. These jeans have the same measurements and parameters as my body does, right? If there isn't a good enough fit across all those variables, then we're gonna reject the null. We're gonna say it does not fit, and we're not gonna buy those pants. Okay. All right, so this goodness of fit test is going to be a way that we're going to compare something we expect, some kind of perfect or model distribution, or something where we have a specific idea of what it should look like, all right, in terms of these categories. So it's similar to the one sample t-test. The one sample t-test, we have a mean. We want to see if it matches to a specified set number that we know ahead of time for some reason. So it'll make more sense when you see an example. And here's our example. So one thing you may already be familiar with is phenotype ratios that when we're doing inheritance and genetics problems, we'll see when we do a Punnett square. So remember that a Punnett square, which is spelled capital P because it was named for Mr. Punnett or Dr. Punnett with 2 N's and 2 T's. We're going to have one parent at the top and the other parent at the side. And we're always doing this just on one parent and their. So we have to know we know the genotype of these two parents. And in rats, white color is recessive. So we have this little a genotype that makes a white phenotype in rats. And we have the brown phenotype, which is the brown allele capital A is completely dominant to the white. So that means if we have, whether we have a big A brown individual, or we have a big A little a brown individual, It's sorry, if we have a big A or an big A little a, it's gonna be brown. Right now, suppose we just have a brown rat and we don't know its genotype, we don't know its parents. So we can do, in this case, a test cross. If we really want now, we probably wouldn't bother to do this with rats, but we might do it if we're breeding dogs and we got a new individual into our population. We don't know its genotype. We can do a test cross it with the recessive, double recessive individual, and see what kind of offspring it makes. And then we can use the ratio of the offspring to determine the parental phenotype, right? So either our brown rat is big A, or it's big A little a, right? If it's big A, all of the offspring are going to be heterozygotes, all of the offspring are going to be brown, right? But if it's a heterozygote, if it's big A little a, then it's going to have big A little a offspring, and it's going to have little a offspring, and it's going to have half and half. All right, it's going to have half and half of each. So we would expect if we cross these two individuals together and our brown rat is in fact a heterozygote, we would get 50% of brown offspring and 50% white offspring. Right. Another way to say this is we expect to get our brown and white offspring in a 1-to-1 ratio. So it's a little easier to think about if we have it in percents. We're going to have 0.5 that are brown. There's different ratios we can have. If it's more genes we're crossing together, we have a bigger Punnett square, then there's lots of different possible ratios. All right, so what are we going to do to test this hypothesis? Well, if we get any, if we get a sizable number of brown offspring, we're going to sort of say, well, if we get any white offspring, we're going to say, well, it's not a homozygous dominant, right? So we're probably going to be testing the hypothesis that it is a heterozygote. So we're going to actually cross the two mice together and say we got 10 offspring, and we found 6 of them were brown and 4 were white. All right, so this is our observed. This is how many babies we saw these rats actually have and what colors they are. We have a total of 10 offspring. All right, now we want to figure out what the expected number of offspring is. Well, we're going to say, okay, suppose I had 10 offspring, right? What's the expected number I should have if they are going to be in this 50%, 50%, or 1-to-1 ratio? All right, so if we have these ratios, the whole, like all of everything together, just like we have 50% and 50% added up together is 100%. Whether we have 2 and 4, or whether we have 2 and 2 making 4, or we have 1 and 1 making 2, we get the same ratio. All right, so we're going to be able to convert a ratio to a percent. We add up. All right, we've got a 1-to-1 ratio. That means we have 2 individuals. So we have to add up the numbers in all of our ratio categories, and then we divide each one by that. So we have 1/2 of brown and 1/2 of white. All right, so that's how we get from the ratio to the percents. All right, so we're going to take that 10 offspring that we want to compare to, because that's how many we have. All right, so we're going to measure this. We're going to count these up. And then we're going to use that number to say I'm also going to have a total expected offspring of 10. And then I'm going to use these percents or proportions in order to calculate if I have 10 offspring, I expect 50% or 0.5 of them to be brown. So I'm going to take 10 and I'm going to multiply it times 0.5. So my expected is going to be 5 individuals, right? I don't want to put 0.5 in here because I need to actually compare a number of individuals, not a percent or a proportion, right? So we have to take the number that we measured in our sample, multiply it by these proportions to get the counts that we would expect to see under the null hypothesis if it was perfect. Right. Because of sampling variability, we may not get exactly 5 and 5 offspring every single time, right? So have we got different enough from 5 and 5 offspring that we would say, whoa, something's going on here. This is not in the proportion that we expect. There's some weird gene interfering with this one. All right, so this step is a little hard to remember how to do it. So just make sure and come back and review this example if you're confused. All right, so let's test hypothesis. So we're going to start off with kind of clearly stating our different scenarios that can happen. If our brown rat is big A, all offspring will be brown. Right? If the brown rat is big A, offspring will be 1 brown to 1 white, or 50% of each color. We need to translate that into a null and alternate hypotheses. And our null hypothesis is going to be our individuals are in the ratio we expect. They match to our model distribution, right? So in this case, our model distribution is what we know should happen based on the principles of Mendelian genetics. All right, so Mendelian genetics and the genotype of the parent is what our null hypothesis. We want to state that. All right. So our null hypothesis is going to be offspring are in this ratio that we expect if the rat's big A, little a. Right. Offspring are or will be 50% brown and 50% white. Our alternate is just the opposite of that. Offspring are not 50% brown white. We can't test the null hypothesis that they're 50% brown and 50% white against an alternate that the offspring are all brown. All right, we'd have to test that separately. All right, so we'd have to do 2 separate tests. These are always just going to be the opposite of one another. Sorry about that. All right, for all hypothesis tests, we're going to set our alpha level, and then we're going to calculate our value of our test statistic. In this case, our test statistic is now going to be the chi-squared. After we calculate the test statistic, we're going to find the p-value using our chi-squared table. And our degrees of freedom for this test is just going to be little k minus 1. So our degrees of freedom is going to be 1 in this case, because if we look back at our observes and expecteds, we've put all of our individuals into only these 2 categories. So little k equals 2. That means our degrees of freedom is going to be little k minus 1 or 1. All right, over on the right-hand side, I've got the equation for calculating the value of the chi-squared test statistic. We're going to sum up for each k our observed minus expected counts. We're going to square that quantity. So in case this is a negative number, that's going to get rid of the negative sign. And we're going to divide by the expecteds. All right, so I'm going to substitute the data from my problem. I've got 6 brown and 4 white rats out of my 10. My expected values are 5 out of 10 in each of the 2 colors. All right, so I've got 6 minus 5, which is 1. I'm going to square that and get 1. All right, so the left-hand term in my sum is going to be 1 divided by 5, 1/5. On the right-hand term, I've got 4 minus 5. That's going to be negative 1. We're going to square it, turns it into 1. And then divided by 5, the number expected. All right, so 1/5. Now, because if things are out of a ratio, we are going to tend to get sort of a balance between how things are out of their equilibrium in one of the categories, and they're going to make up for it in the other. So this is not unusual to get the same deviation for each of our two categories. All right, so quickly you can, these have the same for their expecteds. So you can add the fractions by adding the numerators. So 2/5, or if you get something more awkward, then I divided 2 by 5 on my calculator because I was too lazy to think it through. If you get more awkward fractions and you can't add them because their denominators are different, just convert them into decimals and then add the decimals together. All right, so we got 0.4. That is our value of chi-squared. What do we do next? We go to the chi-squared table and we find where our value of chi-squared is with our 1 degrees of freedom. I'm going to go back up to our table. So we have 1 degree of freedom. We're looking for our chi-squared value of 0.4. Oops, 0.4. Okay, that's less than 1.32. What does that mean? Well, if we go to the top of the table and we find the p-value associated with 1.32 is 0.25, we know our chi-square value is less than our smallest value in the table here. That means if we look at the direction the p-values are going, they are getting bigger towards the left. So our p-value sort of over here to the left of the 0.25 means our p-value is going to be greater than 0.25. All right, so we have our p-value. Our p-value is greater than 0.25. And we picked an alpha of 0.05. Is our p less than our alpha? No, it is not. All right, we do not reject the null. All right, we're not going to reject the null. Our p is too big. It's fairly likely that our null hypothesis matches our data fairly well, right? So we did not reject the null. Let's go back and look at it. What was our null? Offspring are in a 1-to-1 ratio, right? So, or you could say the offspring are not statistically significantly different from half brown and half white, right? It's a little awkward to use statistically significantly sometimes for the results for chi-square tests. All right, so they either are in this ratio or they're not in that ratio, depending on whether we reject or accept or not. All right, now going back one step further, what does that mean? All right, we accepted the hypothesis that the offspring were half brown and half white. So sort of working backwards, we have determined that the brown rat genetically is heterozygote, right? So our brown rat is a big A little a, not a big A big. So in this case where we have data that, fairly obviously matches one of these possible cases more than the other, then we don't have to do both hypothesis tests. Okay, so that's a simple example of a chi-square goodness of fit test, and it is often used to determine inheritance patterns, to determine genotypes in test crosses, like this example that we just did. Don't know the inheritance pattern of maybe a new mutation has arisen in your entire poinsettia nursery, and you don't know its inheritance pattern, then you'll do these test crosses in order to find out, is it dominant, is it recessive, is the dominance complete or not. All right, so lots of lovely genetics use of this chi-square goodness. Another way we can use the chi-square goodness fit test is to do something which is really, really useful for those who are studying evolution. We can do a test to see if a population is in what's called Hardy-Weinberg equilibrium. If you haven't talked about this in a biology class yet, I'm going to give a brief introduction. And then I actually have a separate video that's going to completely walk through doing a full test for Hardy-Weinberg equilibrium with examples. I just want to introduce what it is here and talk about how we're going to apply the chi-square goodness of fit test to it. So if we're talking about population genetics instead of just inheritance patterns, right? So inheritance patterns, we're looking at a single parent with a specific other parent, and we want to know what are their possible offspring and what proportion of genotypes and phenotypes they're going to have. When we're talking about population genetics, we're not looking at individuals, we're looking at the genetic composition of the whole population. So when we talk about the genetic composition of the whole population, it's easier to talk about allele frequencies rather than genotypes or what individuals have a particular genotype. So suppose we have all the individuals in the population are big A, big A. All right, then we have only one allele and its frequency is 1.0, 100% of all the alleles in all the individuals that we have in that population are the same. So there's only one allele and its frequency is 1. Suppose we have equal numbers of big A, big A, big A, and little a genotypes present in the population. Then we're going to have half of all the alleles are going to be big A's, and half of all the alleles in the entire population are going to be little A's. Right, so measuring, like taking all the individuals together and just looking at what alleles they have, and then looking at the proportion of each. We sort of don't care whether they're in heterozygotes or whether they're in homozygotes. All right, so assuming we can know the allele frequencies in populations, and there's ways that we can measure those, and there's no evolution happening. All right, so one simple definition of evolution is that allele frequencies in a population are changing, right? If we've got big A, little a, then the frequency, say, of little a is increasing as we go from generation to generation, right? By natural selection, we tend to have a more favored allele, one that makes more fit offspring. We tend to see it increase as we go from generation to generation. All right, so if we notice an allele frequency changing, then we're actually could be looking at evolution. All right, now there's other ways that it can change that are not natural selection. It can actually be changing if there's other weird things happening, for example, non-random mating, right? So where, there's some individuals with blue spots and other ones with green stripes. They're the same species, but they begin to prefer mating with one another of their same kind. We're going to tend to see that we get more and more homozygous individuals and fewer heterozygotes. So that's a whole other interesting, couple weeks of topic for a genetics class later on. All right, so we can have evolution happening just an allele frequency change. It doesn't have to be by natural selection. It can also be by genetic drift, mutation, or migration. All right, those are biology topics. So I don't expect you to know really anything about them. But if no evolution is happening, then we'll expect to see, we can predict, it turns out, the frequency of alleles in the subsequent generation, just based on the frequency of alleles we have in the current generation. All right, so suppose we have a big A and a little a allele present in our population. That means that, so if we're going to make a Punnett square for population genetics, we want to put the alleles that are present in the population. We aren't saying there's a single individual that has this heterozygote genotype. We're saying I've got some big A's and I've got some little a's in my whole population. right? So whatever you have on the top of your Punnett square, you're also going to have it on the side, because we're assuming that all of these alleles are basically getting mixed together, and they don't necessarily belong together in one individual, right? So the frequency of the big A allele in the population, we're going to call that little p. All right, so big A is an allele. Little p is the frequency that big A allele occurs in our population. All right, so likewise, we're going to assign this variable little q, just by convention, to the frequency of the little a allele in the population. All right, there's no reason that big A and little a have to be in a 50-50 relationship to one another. Right? One of them could be very rare, and it isn't always the recessive allele. We can have a dominant that's actually very rare. Okay. All right, so assuming random mating is happening in our population, no evolution is happening, and that our frequency of our big A allele is little p, and the frequency of our little a allele is little q, then we expect to get proportions of offspring. We put together the big A allele with a big A allele from another individual. The frequency that's going to happen at, this is an and probability question. The probability that we get a big A allele together with a big A allele is going to depend on the frequency. And so we're going to have little p times little p. That's going to tell us the probability or proportion we will expect to have in the offspring of the big A genotype. All right, same way, the probability of getting a little a and getting another little a is going to depend on the probability of getting the little a allele in the first place, little q. So little q times little q, or q squared, is the probability of getting a little a individual in this population. All right, so whether we get a big A from one parent or we get it from the other parent, we'll get a little a from the other one, and we'll have that happen with the probability of little p times little q. But there's two ways that we can get that. So just like when we rolled our dice and we could get a 2 first and then a 3, or 3 and then a 2, and we had to take that into account. We have two ways that we can get a heterozygote. So if we're combining the probabilities of these events happening, right, we can get a big A little a this way, where we got the big A from this parent, the little a from the other parent, or we can get the little a from this parent and the big A from the other parent. All right, so we're going to take this p times q, and we're going to add one of them to the other one and get 2 times p times q to give us the frequency of expected heterozygotes in the offspring. All right, so this idea of Hardy-Weinberg equilibrium in biology is that if no evolution is happening, and we know the frequency of the two different alleles, that we just use the rules of probability mathematics to predict that the frequency or proportion or probability of getting a big A offspring is going to be little p squared. Frequency of getting heterozygotes is 2pq, and the frequency of getting little a is q squared. All right, so we can find the frequencies or the probabilities of our expected genotypes. All right, and we're going to use that information to see if the observed genotypes in our population match to that prediction. All right, so we're going to use this chi-squared goodness of fit test to see if the frequency of our big A's matches to p squared, etc. All right, so I have a second video working through an entire example of how to do that hypothesis test, and that will explain it completely for you. All right, so for now, just know that we're going to use a chi-square goodness to fit test and generally how to do the chi-square goodness to fit test for inheritance problems.